Hi, good morning. My name is Peter Ghoroghchian. I'm the CEO here at Latus Bio. Latus is a 3.5-year-old AAV gene therapy company based in Philadelphia and Boston. We come out of the lab of Bev Davidson from the Children's Hospital of Philadelphia. Really, Latus is a story of trying to overcome challenges that we've seen in the AAV field. Bev, as being one of the founders at Spark Therapeutics and a former president of ASGCT, both on the institutional level as well as in her own lab, has been working over the last decade to try to bridge the gap between what we've seen as the promise of AAV and the reality of first-generation capsids. And so Latus is the beneficiary of a couple things. One is a platform that allows for massively paralleled and unbiased screening of novel AAV capsids directly in non-human primates. And these capsids have all been discovered and then developed with high manufacturability in a GMP-compliant manufacturing process. We also have platforms that allow us to do cell-specific promoter and splice motif design and development, both computationally and experimentally. And Latus completed a public Series A announcement last year in May of $54 million. I'm happy to announce that we have a second undisclosed completion of that part of the financing that we will announce soon, but the proceeds will provide us runway to advance 2 programs through phase 1 studies. Both are slated to begin next year. So at Latus, we're laser focused on solving what we believe to be the #1 challenge with AAV. In 2025, AAV is still probably inarguably the best modality for extrahepatic DNA delivery, but it's fraught with a number of challenges. And each of those challenges, when you kind of zoom higher, really come down to dose requirements. High doses of AAV are what has led to toxicities and patient deaths. It's what has led to manufacturing challenges and a prohibitive cost of goods that's limited the expansion of AAV traditionally beyond ultra-rare disease. So at Latus, our worldview is that the only way to reduce AAV dose is to make capsids that are dramatically more potent and to take direct routes of administration. And to our knowledge, we may be maybe the only or one of a small handful of companies that is evolving capsids via different routes of administration that are optimized for the tissue and disease on a disease-specific manner that's looking to be targeted to find the right cell types and the right tissues. As a result, quantifiably, we have the lowest doses, lowest volumes, and lowest costs in the industry. Our procedure times, whether they're direct CNS administration, intraocular administration, can all be, or even intravenous, dependent on the tissue, can all be accomplished in 2 to 3 hours. Our doses are 1/100th to 1/10,000th the competition. So 1/100th of the competition compared to natural serotype capsids that are directly administered into the eye or brain, and 1/10,000th of IV capsids that are trying to cross the blood-brain barrier. Notably, our capsids are exquisitely cell-specific, so beyond just the target tissue, they've been evolved for particular cell types in that tissue. And to date, in non-human primate studies, our lead capsids have minimal to undetectable off-tissue distribution. And our fully baked-in COGS are, in all cases, less than 10% of the competition, and in many cases are as low as $5,000 a patient. So our business model is really to develop products. Latus has these amazing platforms that allow us to identify capsids and to engineer them. We have undertaken now 10 separate campaigns in non-human primates to identify capsids for different tissues and cell types that are matched to routes of administration that are part of the clinical workflow for that disease. So to date, we have nearly 100 million unique in vivo data points of capsid identity, route of administration, and ultimate distribution, expression, and cell type specificity, offering an unprecedented bank by which we can further do AI-based modeling and refine really the zip codes and street addresses of presumably any cell type in any tissue within the body. What we are focused on, however, is proving this thesis clinically. So we have two capsids, AAV-DB3 and AAV-Appendima, that were recently published. AAV-DB3 in May was published in Nature Communications, and our EP Plus capsid was published in Science Translational Medicine, where we have done head-to-heads against natural serotypes and has shown that these are quantifiably the absolute best capsids in prospectively and then looking at retrospectively against the publicly available data from all of our competitors for targeting these particular cell types and tissues. And we're utilizing them to develop AAV gene therapies for two devastating neurologic diseases. One, an adult neurodegenerative disease, Huntington's, and the second, our Appendima capsid for a rare pediatric neurodevelopmental disease called CLN2, a form of Batten's disease. Notably, these capsids can be used for additional follow-on indications. So our viewpoint is to go after rare diseases where we understand the natural history, where we have biomarkers, where we have established relationships with patient advocacy organizations, where we can run single-arm studies to prove both the safety and efficacy of our capsids, and then utilize them for larger and larger disease indications. I'm going to spend the rest of the talk this morning orienting you to our Huntington's program, both which is our lead and major commercial unlock, but also as a proof of concept for many of the claims that I've just made. So this Huntington's program we call LTS-201. Many of you might be aware, but Huntington's is a familial inherited progressive and fatal neurodegenerative disease. It's very devastating. It affects cognitive and executive function, leading to dementia. It has profound psychiatric effects, leading to both depression, personality changes, substance abuse problems, and also has dramatic effects on motor function, leading to the quintessential choreiform movements, but also in the later stages to dysarthria, dysphagia, bradykinesia, and mutism. Huntington's has been, it's actually relatively, as a rare disease, relatively prevalent. It's 13 in 100,000 as the known prevalence, but recent studies believe that that is underreported by maybe 4x. Huntington has been isolated as a disease involving the Huntington gene, namely on chromosome 4, and it is due to a repeat expansion of 3 nucleotides, CAG, in exon 1 that lead to an abnormal protein that has a plethora of different biological effects, ultimately leading to neuronal death. So Huntington's is a 2-hit phenomenon. You inherit 1 copy of mutant Huntington from mom or dad, where you have more than 39 CAG repeats in exon 1 of Huntington. And for reasons not completely known, you have this mutation everywhere in your body, but only within the medium spiny neurons of the caudate and putamen, as well as in cortical projection neurons, and to a lesser degree in the liver, do you have a decades-long somatic repeat expansion. So you inherit 39 copies at birth, but over decades, the length of these CAGs increases in those particular cell types and in those tissues. And the manifestations are entirely in the brain. So these are a neurodegeneration and death. So there have been, to date, no approved disease-modifying therapies for Huntington. Really, the field has been reinvigorated by the recent success of Unicure and AMT-130, which for the first time has shown disease modification, bringing new hope for Huntington's disease patients and offering a paradigm of comparison about how we pursue LTS-201 versus the new bar that's been set and the pioneering work that Unicure has done in AMT-130. So there's a lot of interesting avenues for targeting Huntington's disease. So AMT-130 is an engineered microRNA against wild-type Huntington delivered with AAV5. There are a number of other Huntington-lowering approaches, most of which target both wild-type and mutant Huntington using small molecule splice modulators, antisense oligonucleotides, and siRNAs. So we believe that LTS-201 has a differentiated profile, namely that we're offering a better target, a better capsid, and a better delivery proposition than the competition. So our target, instead of lowering both wild type and mutant huntingtin, as does most other modalities, we're targeting a different target, MSH3, which is a DNA repair enzyme from GWAS studies, human postmortem tissue analysis, mouse genetic and pharmacologic studies has validated that MSH3 is the causative protein in the somatic repeat expansion of Huntington. So we are specifically engineering a microRNA to knock down MSH3 only in medium spiny neurons where the disease pathology occurs, and in an allele-specific fashion. This should only work on reducing the expansions of mutant huntingtin without affecting wild-type huntingtin levels, which is thought to be toxic. One of the ways that we're able to achieve this is our capsid. AAV-DB3 shows a vastly superior brain transduction profile compared to AAV5 in mice and in NHPs. So the micrographs that I'm showing you in the middle panel, let's see if I can, these are hundreds of fluorescence images that have been stitched together to create a coronal cross-section of the caudate and putamen of a rhesus macaque that was treated in one hemisphere with AAV-DB3 and in the contralateral hemisphere with AAV5. What you can probably appreciate from these low resolution, low magnification images is a tremendous transduction profile throughout the entire caudate and putamen. When quantified at high doses, this is 99% of all medium spiny neurons in the caudate and putamen with AAV-DB3. And in the same contralateral, and same animal in the contralateral hemisphere, AAV5 resulted in less than 1% transduction of MSNs. The last piece is delivery. Because of this potency and transduction profile, LTS-201 can be administered by a single hemispheric injection, in contrast to AMT-130 that requires 3 injections per hemisphere, 6 total injections over 15 to 20 hours. LTS-201 can be administered in 2 hours using anywhere from a 1 log to a 2 log reduced dose with no convection enhanced delivery, and results in no MRI abnormalities in the post-op MRIs in these NHPs. LTS-201, meaning AAV-DB3 delivering a engineered microRNA against MSH3, has been shown in the Q111 mouse model of Huntington's disease to result in reduction of MSH3 protein and a commensurate reduction in somatic instability within the mouse model. Importantly, in non-human primates, we've conducted an NHP dose range finding study where we've looked at 3 separate doses and looked at RNA-scope fluorescence microscopy to quantify MSH3 levels within medium spiny neurons as a product of escalating doses of LTS-201. What we see on the right panel is a deep, dose-dependent knockdown of MSH3 in all medium spiny neurons within the adult rhesus macaque brain, achieving upwards of 90% knockdown of MSH3 in 100% of MSNs. To our knowledge, this is the deepest knockdown of the most important target of the right cell type in the right substructure to impact Huntington's disease. We've also developed proprietary modeling that, based on using postmortem samples from patients that can predict the rates of somatic repeat expansion based on MSH3 levels, as well as bringing in clinical data from the Enroll-HD and Track-HD database. This allows us to predict the efficacy of our gene therapy based on the number of medium spiny neurons, the depth of MSH3 knockdown, the age of the patient, and the number of CAG repeats. What this has shown that is that at the low dose, where we're achieving 50% knockdown of all medium spiny neurons, this would predict more than 5 years of clinical benefit for the vast majority of Huntington's disease patients. At the high dose, where we're achieving upwards of 90% MSH3 knockdown and 99% of MSNs, this would equate to more than 50 years of clinical benefit for all HD patients. So importantly, what you want to do is be able to identify and utilize surrogate biomarkers in order to understand activity in low-powered and early-stage studies. And Huntington's has two of these biomarkers. One is neurofilament light chain, and the second is caudate volume MRI. So we have shown that we have very small spikes in neurofilament light chain compared to AMT-130, and specifically that we have no changes in the post-procedure MRI, allowing us to follow both NFL and caudate volume for early readouts. So this just shows our timelines. We're tracking for 2 INDs, one that we are working through in Q4 of this year in a earlier disease in CLN2, and the second one targeting an IND in mid-2026, with both phase 1 studies commencing in 2026. Thank you very much.